off the beginning, so I don't want to lose any information. So Israel, let me know that you're just going to introduce yourself and then dive right in. If you want to redo any slides, just do me a favor and say, I'm going to redo that, and then start from the top of that slide, but give me a couple of seconds before you start talking again, just for editing.
Okay.
Other than that, I'll be here on mute, just taking notes. If there's no edits, that's great. And whenever you're ready.
Okay. Yeah. Just as a heads up, for some reason I've had a dry cough like yesterday and today, and if I end up having to, I have a glass of water here, a bottle of water here. If I end up like coughing away, I'll cancel the slide. Take a
no
drink.
Worries. If it takes if it takes you, just take a moment and we'll start from the top of that. Don't worry about it.
Okay. And finally, overall target time is around 45 minutes, I was told. 45, 50 minutes?
Yeah, that's the goal.
Great. Okay. Perfect. And are you going to cue me when it's recording?
Oh, it's been recording. So it's really just whenever you're ready.
Sounds good.
Okay,
ready? 3, 2, 1. Hi, I'm David Shaffer. I'm a professor of chemical and biomolecular engineering, bioengineering, and molecular and cell biology at the University of California at Berkeley. And today I'm going to be presenting to you some of our work on the use of directed evolution as a platform to engineer improved gene delivery systems for clinical gene therapy. The motivation for all of this work is that regardless of which tissue you look at within the body, there are unfortunately a large number of chronic progressive degenerative disorders that will kill off tissues and cells inside the body, rob people of their quality of life, and eventually, in some cases, their life. So just as an example, if you take the nervous system, we have complex disorders such as Alzheimer's and Parkinson's disease, as well as monogenic disorders where you can pin the blame for the disease on specific mutations within genes in the genome. Regardless, in either of these cases or categories, we need new therapeutics. So if you take a step back to a central tenet of biology, essentially the central dogma, we know that information is stored at the level of DNA, transcribed into RNA, translated into protein, and that while DNA is the storage medium for that information, RNA and proteins make up the functional molecules that are responsible for the everyday function of our cells. So from the point of view of a drug developer, each one of these levels of molecules represents a drug target. The majority of drugs that are currently used in the clinic target at the level of proteins. So we have small molecules that bind to, typically in the active site of an enzyme, to inhibit its function. We have monoclonal antibodies that bind to particular protein targets outside of the cell and often inhibit their function. We know that RNA itself can also be a drug target. We have several FDA-approved RNA interference drugs that degrade messenger RNAs in a targeted fashion. We also have antisense oligonucleotides, and we know as well from the COVID vaccine that messenger RNA itself can be a therapeutic molecule. What ties together the research in our lab, though, is that we drug at the level of DNA. If you'd like to add a new gene to the genome, that's a gene therapy. If you'd like to change the sequence of an existing gene, that's an edit. And finally, if you'd like to add an entirely new gene to a tissue, that's a cell therapy. But what these share in common is that they target at the level of DNA. DNA is the therapeutic modality. And one advantage of that, in some cases, is the pharmacokinetic half-life of DNA. So just as an example, if you take the lifespan of somebody who's been diagnosed with Parkinson's disease before they unfortunately pass away, it's in the order of 15 to 20 years. Congestive heart failure, as well as age-related macular degeneration, typically the remaining lifetime of the patient diagnosed is around 10 years. The pharmacokinetic half-life of the small molecule is in the order of hours. If you move to a monoclonal antibody, it increases to days to weeks, perhaps. However, you can see that by definition, for long-term chronic disorders, these two therapeutic modalities are treatments. They're not cures. DNA, in the form of a gene therapy, however, has the potential to persist for the life of an entire patient. So, for example, here are two clinical trials which we'll be talking about. Both have resulted in FDA-approved therapies where patients were dosed back in the 2000s, and after that single administration, they're still enjoying therapeutic benefit today. So one of the major challenges, in fact, actually the Achilles heel of the gene therapy field has been delivery. So specifically, you can identify a DNA medicine that could potentially treat a disorder, but how do you deliver enough of it into enough nuclei of a target tissue to begin to modify disease progression. So for this, the field has, over the past several decades, relied upon viruses, since viruses deliver nucleic acid as part of the replication cycle, and you can trick a virus into delivering a therapeutic piece of DNA rather than its own genome. And the most successful virus to date for in vivo gene therapy has been adeno-associated virus, or AAV. So AAV's genome is quite simple. It's composed of only 2 genes. The first one, called Rep, encodes some enzymes that are responsible for mediating the replication of the viral DNA inside of a cell. The second one is the focus of roughly half of my career, and that is the capsid gene, or cap gene, which encodes structural proteins, 60 copies of which will self-assemble to form this icosahedron, this hollow soccer ball, if you will. So the way that it works then is one of the copies of the Rep protein will dock at a pore on the surface of this soccer ball, go and grab a single-stranded genome of the virus and pump it into the interior of the particle to be able to create an infectious variant. And these viruses have some interesting attributes. Among them, they're among the smallest of all viruses, whether it be eukaryotic or prokaryotic. They're non-pathogenic, have never been associated with human disease. And a number of natural variants have actually been isolated, and they contain on their surface some variability in amino acid composition, which results in some changes in infectious or delivery properties, and raises the interesting possibility that by engineering the exterior of that soccer ball, you could create an efficient targeted gene delivery vehicle. So the way that you convert a natural virus into a recombinant virus or vector is by simply deleting the 2 viral genes from the viral DNA, the Rep and Cap gene, and placing them onto a separate piece of DNA, which then liberates space for the insertion of, in principle, any gene of interest, provided that it's small enough to fit inside the virus, and then the soccer ball in turn becomes loaded with a copy of the gene of interest, the therapeutic DNA, rather than its own genome, and will function like a Trojan horse to deliver the therapeutic DNA rather than the viral genes. So these particles have a number of interesting properties. They're relatively safe. It turns out that 90% of the human population has already been exposed to one or more natural versions of AAV, and again, never even noticed it because it's non-pathogenic. So that's a very safe starting point for use as a therapeutic. These viral particles, although they evolved for infection of lung, are somewhat efficient in gene delivery to a number of other tissues and have been, as a result, used in clinical trials for muscle, liver, lung, brain, and retina. Upon depositing the DNA cargo in the nucleus of a target cell, that cargo can persist for long periods of time, provided that the cell is not dividing, potentially even for the rest of the lifetime of a patient. There still needs to be progress in manufacturing at scale, but they're good enough to be able to get into the clinic. And as a result of all these attributes, 6 AAV-based gene therapies have been approved by the FDA to date, including some that we can see over here. So the process of a gene therapy is pretty complex in that we have this 4 million molecular weight particle. It's produced in a mammalian cell process and then administered into the body, such as intravenously or through a direct injection into a tissue. In the case of an IV injection, it has to circumvent the bloodstream and be able to find a target tissue of interest, such as the heart, extravasate through the blood vessels, end up in the interior of the tissue, penetrate that tissue deeply, find a receptor on the surface of a target cell, get taken up by endocytosis, escape the lipid bilayer from the endosome into the cytosol, traffic to the nucleus, and then deposit the DNA inside of the nucleus. So this is a complex drug product, and as a result, there are multiple layers of challenges associated with developing these for the clinic. The first of these is really going to be the focus of my talk today, and this is by and large because it's been the major focus and challenge within the field over the past several decades, and that's delivery. That getting these pieces of DNA into enough nuclei of enough target cells is a very challenging process given all the barriers along the way, and as a result, efficient delivery of therapeutic levels of the transgene has been the major challenge. We also have a manufacturing challenge. Now that there's 60 FDA-approved gene therapies and more on the way, it's challenging to produce enough of this material to economically treat large patient populations. I'm not going to be talking about that today, and in addition, I won't be discussing the third challenge, which is the regulation challenge, that after this DNA has been deposited in the nucleus of a cell, there's a promoter inside of that DNA that regulates how much, when, and where an RNA is expressed. And in some situations, we very tightly need to control that in order to have efficacy without any sort of off-target effects. And so this is increasingly going to become a challenge in the field. But today I'll be talking about delivery, as I mentioned. So what are the barriers to efficient delivery? The first of these we effectively already talked about, which is the fact that greater than 90% of the human population has been exposed to one or more natural versions of AAV, and as a result of this accidental vaccination, we have high titers of antibodies directed against them in many bodily fluids throughout the human body. These viruses are typically inefficient at biodistributing throughout the body. They're, in many cases, efficiently taken up into the liver after an IV administration. Doesn't mean they infect the liver that well. They simply are depleted from the bloodstream by uptake into the liver. And as a result, they biodistribute to other tissues rather poorly. There's inefficient spread within a target tissue. The natural versions of AAV have zero capacity to target delivery to some cells versus others. They're often very inefficient. This is a respiratory virus and not a very infectious one at that. And if you showed a cardiomyocyte or a photoreceptor neuron, it's not prepared or has not been prepared by evolution to be able to infect those cells. And in addition, lastly, as I mentioned, they're hard to manufacture. So this naturally begs the question, how is it that we have FDA-approved gene therapies at all based upon AAV? And the answer to that is that these therapeutics to date have really brute forced the delivery, and they do that either with exceedingly high doses or with invasive routes of surgery to place the virus right next to the target cell. So this can work for a subset of human diseases, but it really begins to fall short for other situations, in fact, the majority of human disease. So for example, let's say you would like to treat a condition that affects a tissue other than the liver, perhaps muscle. So one way in which you can do this is to take an AAV and simply increase the dose to the point where a sufficient amount arrives on target at muscle. This has actually been done in human clinical trials, for example, by a company, Astellas, that was trying to treat a fatal disease that affects young boys, a muscle degenerative disease. And they simply increased the amount of AAV8 that was administered to the patients. And even though, 999 particles arrived at the liver for every one that arrived at muscle, they could stoichiometrically increase the amount that was administered to the body to get a good amount that arrives on muscle. But to do this, they had to increase the dosage to over 100 AAV particles for every single cell within the body. So AAV is non-pathogenic, it's quite stealthy, does not typically elicit a strong immune response, but if you're administering that much material, 65 milligrams of virus to a patient, the immune system begins to notice it. And unfortunately, this led to significant adverse events, immune responses to virus that had been delivered or off-target localized to liver. And this led to several deaths within a clinical trial. So the bottom line is that nature never evolved viruses for our convenience to use as medicines. So it's up to us to re-engineer these soccer balls, these delivery vehicles, at the molecular level to make them much more efficient and much more targeted for our tissue of interest. So that's extremely difficult to do because, as we talked about, this is a very complicated delivery process. So viruses are very complicated, and this is a 4 million molecular weight, 5 million if you count the DNA, product that's the product of millions of years of evolution. And more often than not, when we tinker with it and try to change its delivery properties, we simply crash its ability to deliver DNA at all. So we began to work on this problem about 25 years ago and developed or conceived of applying a new approach to be able to re-engineer AAVs for much more efficient and targeted delivery to a given cell or tissue. And we adapted this approach that had been just started to be applied to enzymes, approach called directed evolution, and became the first to apply it to AAV. So the way that this approach works is that we can take a natural version of AAV, a wild-type capsid, which is a useful or interesting starting point for evolution, and we simply quite simply emulate the way that nature genetically diversifies genes, which is through mutation and recombination. So we apply a variety of molecular biology approaches, typically relying upon PCR, DNA synthesis, and cloning to generate enormous libraries of these AAVs. So most recently, we've actually been using computationally guided library design, as shown in some work that we published earlier this year, in which we used machine learning to make more effective libraries. But the bottom line is that we have over 40 libraries. Each of those has on the order of 10 to 100 million variants. So in sum total, we have over 1 billion different variants of AAV. So we then take these candidate genes encoding the viral capsid, package them into particles such that each particle is composed of a variant capsid, a variant soccer ball, if you will, surrounding the DNA that encodes that capsid. So we can then, for example, say that, let's say this particular variant within this haystack, this needle is the one that is really effective at mediating delivery to the heart after intravenous administration. We can take this entire library in a syringe, apply it intravenously to a model, initially mouse, then dog, pig, and more recently primate, then isolate the target cell, such as a cardiomyocyte, amplify the virus that made it to that cell, repackage it, re-administer it, and go through these successive rounds of selection such that we end up converging on the winner, the variant that has the best properties for delivery to the heart. And of course, at every step along the way, we can pop open the genome, sequence them, and recover the capsid sequence, the genetic information that's responsible for this improved delivery. So I'd like to talk to you about several applications of this technology. The first of these is I'm going to show you an example within mouse. The second is I'm going to move on to our ongoing efforts to advance this into human clinical trials. And then I'm going to talk about the fact that directed evolution is a general platform that can be applied towards engineering better properties to other gene delivery vehicles. So the first tissue in which we worked was the retina. And this is the structure of the retina, which you have, of course, on the interior of the eye, a lens. You have some gelatinous vitreous fluid on the interior of the structure. And on the back of the eye, there are a number of layers of neurons. All the way at the back is the photoreceptor layer that responds to a photon by firing an action potential to excite a number of layers of interneurons. And then finally, that information reaches the ganglion cell layer and is propagated along the axons of the GCL, which bundle to form the optic nerve and convey that information into the brain. So unfortunately, mutations in over 200 genes can result in progressive degeneration of photoreceptor neurons. So think retinitis pigmentosa or Leber's congenital amaurosis. In addition, more complex diseases such as age-related macular degeneration can also kill off these photoreceptor neurons. So it'd be desirable to deliver a therapeutic piece of DNA that can target these photoreceptor neurons and protect and repair their function. So the most convenient or the most logical way to administer a therapeutic into the eye is with a so-called intravitreal injection, in which a needle is gently drawn through the sclera and injected into the vitreous humor, the liquid of the eye. So this is very, very safe. It's actually done millions of times a year in the United States to deliver proteins that inhibit vascular endothelial growth factor in patients with wet AMD. In addition to being a very safe approach, it also gives the therapeutic the opportunity to come into contact with the full surface area of the retina. Unfortunately, this does not work for natural versions of AAV because between the vitreous of the eye and the photoreceptor layer, the target of gene delivery, lies a couple hundred microns of dense tissue, and this prevents the passage of natural AAVs. So to be able to overcome this, investigators have invented a new surgical route of administration called a subretinal surgery, in which the needle is actually punctured through the sclera, then extends all the way to the photoreceptor layer, or to the retina, punctured through the retina, and injected behind the photoreceptors and between them and the underlying retinal pigment epithelium. And this is problematic for a couple of reasons. It's forcing the photoreceptors away from the underlying cell layer, which by definition is a retinal detachment. And so for patients who are already suffering from retinal degenerative diseases, this can be traumatic and add insult to injury. And second, you can't really control laterally where the fluid flows away from the needle tip. So you typically end up with a random shape of transduced cells. And in addition, it's only really protecting around 5 to 10% of the surface area of the retina. Yet this was good enough for an FDA-approved gene therapy, actually the first FDA-approved gene therapy based upon AAV, which is currently being distributed by Roche for the treatment of a retinal degenerative disease called LCA2. But we wanted to ask, can we actually do better? Can we engineer AAVs that are capable of making it to the outer retina, to the photoreceptor layer from the vitreous? So to address that, we took our library, we injected it into the vitreous initially of a mouse model, waited a couple of weeks for the winners to be able to reach the photoreceptors, harvested the photoreceptors, re-administered the pool, and went through this 5 times, or 6 times rather, so that we ended up converging down to a small number of variants that were dominant within the resulting pool. 2/3 of this pool was composed of a single variant, and when we developed this cartoon of its structure, you can see that it has sequence diversity shown in red point mutations as well as green peptide insertions that are on the surface of the virus and in a place where they could potentially make a difference in the way that the virus interacts with cells and tissues. To test the delivery property of this virus, we simply loaded it with DNA encoding the green fluorescent protein and administered it to the mouse eye. This is the parental virus, AAV2, as a control. And you can see that there's barely any AAV that reaches the photoreceptor layers, essentially barely reaches any layer within the retina, upon administration into the vitreous fluid, which is at the bottom of the slide. In contrast, if we administer the same dose of the engineered AAV, you can see it's reaching every cell layer from bottom to top. I'll mention as well that AAV2 is actually the version of the virus that's used in that FDA-approved gene therapy by Roche. And quite clearly, they had to do a subretinal surgery to get the virus where it needed to be. But potentially with an engineered virus, we could do a much simpler administration into the vitreous and benefit from the fact that that virus will deliver across the entire surface area of the retina, unlike a subretinal surgery where it only delivers to 5 to 10% of the surface area. So we analyzed the mechanism by which this virus is making it deep within the eye, and it turns out that natural AAVs bind to proteoglycan, such as heparin sulfate, with extremely high avidity. And there's a lot of heparin sulfate within this first layer of tissue between the liquid of the eye and the retina. And as a result, AAV sticks like glue to that first layer and doesn't have any opportunity to penetrate. So some of the modifications to the viral capsid actually reduced its affinity for heparin sulfate, and this actually resulted in better penetration deep into the retina. However, these particles were also endowed with the ability to bind to a novel receptor that we've not yet identified. So I'd like to present application to another tissue of interest, which is the brain and the spinal cord. And these are very large, delicate tissues that it's very hard to deliver DNA to. And as an example, within a somewhat recent clinical trial, investigators tried to deliver across the entire large volume of the brain to be able to treat a lysosomal storage disease called Sanfilippo B. So they did 8 drill holes and 16 injections into the brain, and this was still, insufficient to space fill that large tissue and was unable to modify disease progression. So we noted a number of years ago that there's an interesting mechanism of AAV transport within central nervous system, which is that if you deposit AAV near an axonal terminus, it can actually get endocytosed and transported retrogradely along that axon back to the cell body, so you can infect a cell from a distance. So this can be used in a couple of ways. That if you're trying to infect a large brain region, where it'd be very difficult to do that with a lot of injections, but a number of cells or neurons within that region send their projections to a small focal location, you can do a single injection of that focal location and have the virus retrogradely spread back to that large structure. In addition, in situations where you're trying to infect a motor neuron, such as to treat spinal cord disorders, you can actually do the injection into muscle and enable retrograde transport along that motor neuron to be able to correct the muscle disorder. So this was done in a mouse model of amyotrophic lateral sclerosis. So unfortunately, natural versions of AAV are very inefficiently retrogradely transported, and a company that tried to advance this into the clinic was not successful as a result. So we wanted to engineer a retrogradely transporting AAV, and layers 2 and 3 of the cortex send their projections to a small region called the pons. So we could inject the library at the pons, wait for the virus, the successful virus, to get retrogradely transported to the cortex, isolate the cortex, isolate the AAV genomes that made it there, re-administer the pool, and go around that loop a number of times until we converged down to a single variant, which had, again, some surface diversity in a way that could potentially alter how it interacted with cells and tissues. We loaded this virus with cDNA encoding TD tomato, injected into the pons, and you can see it lights up the entire cortex. We wanted to quantify this more effectively, and so we ended up using a situation in which GFP was nuclearly localized, so we could quantify the number of cells that took it up. You can see that AAV2 barely hits the cortex. You can't see really any green cells at all within the cortex after injection into the pons. Another version of AAV, which is an engineered one, shows a mild speckling, but our variant engineered for retrograde transport, AAV2 retro, is actually 50-fold more efficient than any natural version of AAV that we tested. So this became sufficiently efficient that with a small number of injections, such as to the superior colliculus, the thalamus, and the pons, we could actually get retrograde transport to very large volumes of brain tissue. So we wanted to utilize this to be able to deliver a cargo. And of course, CRISPR-Cas9 is a very interesting or promising cargo. And I joke that because I work at UC Berkeley, I'm contractually obligated to use it within my lab. So we wanted to genetically modify a region of cells by using retrograde transport of CRISPR-Cas. So specifically, we utilized a strain of mouse that was expressing TD tomato as a transgenic mouse within the cortex, and we loaded inside of the AAV a Staph aureus Cas9 armed with the guide RNA to cut or to knock out the TD tomato gene. So we then injected this with the targeted guide RNA or an irrelevant control RNA into the pons of these animals, allowed it to become retrogradely transported to the cortex, and found that within the cortex at a distance, the SA Cas9 was able to knock down over 95% of TD tomato expression from that long distance within the large volume of the cortex. Okay, so we have since used this in a couple of ways to be able to deliver CRISPR-Cas9 to achieve efficacy within animal models of human disease. So I'm not going to go over these in much detail because they've already been published, but specifically we show the first CRISPR-based efficacy in an animal model of a neurodegenerative disease for amyotrophic lateral sclerosis, and we also show the first CRISPR-mediated increase in the lifespan in an animal model of Huntington's disease. But I'd like to pivot now to our efforts to be able to get this technology into the clinic. And to do so, we wanted to translate it into a company that could raise the capital, quite frankly, to be able to advance this into human clinical trials. So back in 2013, I co-founded a company called 4D Molecular Therapeutics, which is located a few miles from the Berkeley campus. This is now a public company with on the order of 150 employees that has advanced this technology into 3 therapeutic areas: the retina, the lung, and the heart, and actually has, this is a bit out of date, 6 ongoing human clinical trials and 1 open IND. So I'm going to tell you about 2 clinical programs within the company. This is all publicly available information on the company's website, but the first of these is focused again on the retina. So it is quite likely that a vector evolved for optimal delivery to a rodent will be suboptimal within a non-human primate, and we actually found that. And since we want this vector to be efficacious within a human, and we felt that the non-human primate was the best preclinical model to prepare a virus to work within a human clinical trial, we repeated the evolution within the primate. So this resulted in a variant of AAV that was capable of very efficient delivery to the outer retina, to photoreceptors upon administration into the vitreous. And I'm not going to show those data. Again, it's available on the company's website, but I wanted to jump ahead to some reported data for the application of this vector to be able to treat a condition which is quite prevalent in the United States, specifically age-related macular degeneration. So you'll remember I mentioned to you earlier that protein therapeutics for wet AMD are actually administered to patients millions of times a year. And for example, this particular protein called aflibercept, which is commercialized by Regeneron, is an inhibitor of vascular endothelial growth factor. And by getting injections of this therapeutic, patients with wet AMD experience a slowdown or a halting of the progression of wet AMD within their retinas. Unfortunately, proteins have a limited pharmacokinetic half-life in the body, as we've discussed, and patients who have wet AMD typically have to get anywhere from 6 to 8 to 10 to 11 injections of this protein therapeutic per year. So getting 1 shot in the eye is quite acceptable. Getting 10 a year for the rest of your life is something that is a bit difficult to take. So typically patient compliance goes down, and as a result, patients begin to unfortunately experience disease progression and loss of vision. So we ended up generating a version of this AAV vector that was optimized for the retina, which we call R100, and loaded it with cargo to be able to combat wet AMD. So the most active ingredient is protein that encodes this DNA that encodes this protein that neutralizes VEGF. So we quite simply took this product and vectorized it, encoded it as DNA inside of this vector. It was tested preclinically within an animal model of wet AMD, in which a mild laser illumination is used to be able to create a slight burn in the back of the retina, and this results in the ingrowth of vasculature into that region. And so you can administer an AAV encoding an anti-VEGF and determine whether or not that can inhibit the formation of these lesions, of the growth of vasculature into these regions in the eye. And this is a 6-month in-life study that also tested the safety of this therapeutic. So you can see at 3 different doses, and trust me, these are actually very low doses of AAV. By comparison, the amount that was administered to those patients with that muscle disorder was on the order of 10 to the 16th or greater. So this is on the order of 10 to the 11th per eye. It resulted in expression of therapeutic amounts of this protein. And you can see that it completely inhibits, even at the lower dose, the formation of these vascular ingrowths or these lesions within the eye. So based on these preclinical data, this therapeutic was advanced into a phase 1 and then a phase 2 clinical trial. The phase 2 data have been reported several times this year. This is the initial reporting of the data to a 51 patient broken into 3 cohorts. A control cohort, and this cohort that received a single intravitreal dosage of 3 times 10 to the 10th, which is actually 3-fold lower than the lowest dose administered to the non-human primate. These were patients who actually had a very high treatment burden, meaning that they required 10 injections of the anti-VEGF protein per year. So upon injection, there was no clinically significant intraocular inflammation, and patients were tapered off of prophylactic steroids within 26 weeks. And as you can see here on the left-hand side, each one of these dots represents the injection of a protein inhibitor of VEGF. But after the administration of the AAV, you can see there was a substantial reduction in the need for protein therapy, meaning that the vision of these patients, as well as swelling within the retina, were stabilized throughout the course of this study, such that there was a 90% reduction in the need for injection of protein therapies. So this is an ongoing study and will actually initiate as a phase 3 early next year. So I'd like to very quickly talk about cystic fibrosis, and this has been the target of over 20 gene therapy clinical trials, which to date within the field have all failed due to a lack of delivery. So we evolved a variant of AAV for efficient delivery to the lung in a non-human primate. Through aerosolization and administration to the lung and recovery of AAV that made it to the epithelium. We took this variant, embedded inside of it, DNA encoding the CFTR, or a truncated version of CFTR, which is the gene that's mutated within cystic fibrosis patients. And I'll skip this really quickly for the interest of time. This is non-human primate data where brown immunostaining shows that there was successful delivery to the trachea, bronchi, and alveoli within these subjects compared to a negative control, vehicle-only control. This was translated into a human clinical trial, and this is from the first cohort of patients, n is equal to 3, in data reported last year. This is control lung tissue, in which you can see there's no immunoreactivity, whereas in a number of biopsies from patients who received the vector, greater than 98% of the cells were expressing the CFTR. And this led to at least qualitative, not statistical yet, but encouraging results in which there was a stabilization or a slight improvement in the lung volume, the lung function, the volume of air that could be exhaled from the lung in a second, as well as an improvement in quality of life. So the company now has 6 ongoing human clinical trials, 4 in the eye, 1 in the heart, which I didn't discuss, as well as 1 within the lung. But for the remainder of the talk, I'd like to discuss, again, going back to basic vector development, the creation of a new technology to enable directed evolution of other viruses. And this is a technology that is based upon CRISPR. So the application here is not a rare monogenic disease, it's actually cancer. And there are, as we well know, a number of successful cancer immunotherapies that have come into the market in recent years and have really revolutionized the treatment of malignancies. So these include both CAR T therapies, in which a T cell is armed with the capacity to recognize an antigen that is selective within a tumor cell, and so the T cell can attack the tumor, as well as checkpoint inhibitors, which stimulate immune responses, both adaptive as well as innate, against the tumor. So while these have been successful and have resulted in a number of FDA approvals, they do have limitations. And specifically, there has to be some basal level of immune surveillance within the tumor for these therapeutics to succeed. And so they work on quote-unquote hot tumors that do have some immunological presence, but in cold tumors that lack any sort of basal surveillance of the malignancy, they're not very effective. So there's a low response rate in cold solid tumors. So one promising approach to address this problem is an oncolytic virotherapy, which is a virus that has been engineered to selectively replicate in and deliver therapeutic cargoes to tumors. There's been a lot of momentum within this field over the past few months or years, including, for example, very high success rates reported by Candle within a phase 1 clinical trial, as well as CG Oncology, which has successful clinical data that actually fueled the very first biotech IPO of this year. So these successes rely upon the fact that these therapeutics have the potential to combine the best of both worlds for conventional cancer immunotherapies. So by replicating within a tumor selectively, a virus actually presents multiple antigens for the immune system to come in and recognize the tumor. In addition, these viruses can express one or more immunostimulatory cargoes that are encoded within their DNA that can actually result in further boosting of immune responses of the tumor. And all of this is in an off-the-shelf product. However, there's a significant limitation, which should be quite familiar to all of us, especially based upon what I discussed for AAV, which is delivery. So in this case, the delivery problem is threefold. It's challenging to be able to get enough of the virus to go from the point of a needle tip of a syringe to the tumor. Ideally, we would like to do that intravascularly. It's challenging for the virus to replicate and spread on the interior of the tumor. And finally, ideally, we would like the virus to actually take hold within a primary tumor, become released as a biofactory from that primary tumor into the vasculature, and be able to home to and find secondary tumors to eliminate them. And that intertumoral delivery is also challenging. So our goal is to really engineer better oncolytic virotherapy delivery to unlock the full potential of this therapeutic for cold solid tumors. So I described to you earlier that directed evolution is a very powerful engine to be able to create better virotherapies. Specifically, we showed that with AAV. However, there's a challenge when applying directed evolution to another virus, such as a large virus, and that comes about with the genetic diversification. I mentioned to you that we genetically diversify AAV based upon standard molecular biology techniques, such as DNA cloning, PCR, DNA synthesis, but oncolytic virotherapies are typically much, much larger than AAV. So for example, I'm going to be talking about a vaccinia, which is 190 kilobases compared to the 4.7 kilobase genome of AAV. So the standard molecular biology techniques that we can use for genetic diversification and creation of a library of AAV quite simply don't work for an oncolytic virus such as vaccinia. So fortunately, a few years ago, with the collaborator John Duber, we engineered another approach based upon CRISPR to genetically diversify a target locus within a genome. So the way that this molecule works, and it's called Evolver, is that we fuse a nicking Cas9 with a error-prone DNA polymerase. And then you can use a guide RNA to recruit this molecule, Evolver, to, in principle, any locus within any gene within any genome. And the guide RNA then enables the Cas9 to introduce a nick into the target strand. The Evolver dissociates, and then the DNA polymerase engages with that nick and lays down or translates a stretch of error-prone DNA synthesis. And so this can be a way to genetically diversify a target locus within a complex genome. We've shown it works in bacteria, yeast, human genome, as well as viral vectors, namely vaccinia. And the reason to do this in a targeted locus fashion within vaccinia is that the majority of the viral genome is not associated with delivery, and mutations in most places within the genome are actually going to harm the virus. So we want to localize genetic diversification only to regions that can make a difference for delivery. So we now have the capacity again to genetically diversify, and we can put this together with selections within cells or tissues to actually have a mechanism for continuous genetic diversification and selection for better delivery to tumors. So our substrate for this evolution is vaccinia, and it checks off a couple of boxes to be used as a therapeutic. So first of all, there is safety. The fact that vaccinia has been used actually as a vaccine for smallpox and administered to billions of people worldwide. So it's a very strong safety record. And furthermore, it's possible to make genetic mutations or deletions inside the virus that render it capable of only replicating inside of a tumor. Second, manufacturability was worked out as a result of vaccinia being a vaccine. In addition, there's a really interesting form of vaccinia called the AAV form, or extracellular enveloped form. So this wraps an extra layer of membrane around the viral particle and makes it extremely stealthy. So it's able to evade the immune system, antibodies or complement. And as a result, this form can be administered or is effective intravenously. And we think that this is going to enhance the efficacy of this particle. And finally, there's the fact that we have the capacity to encode or to embed over 40 kilobases of therapeutic cargo inside of the virus to express immunostimulatory molecules inside of the tumor. I'm pausing for a second here because I have a low battery signal on my computer, so I need to be able to plug it in. And I'll move on to the next slide in just a second.
Not a problem, David.
Okay, great. 3, 2, 1. So shown here is data in which we actually applied a Volver to genetically diversify and select vaccinia virus for improved delivery to tumor cells. This is an initial proof of concept study that was done in cell culture using a human colorectal cancer cell line. So shown here on the left top panel is on the x-axis nucleotide position within the vaccinia genome. And this is a locus within the vaccinia genome that encodes a surface protein on the AAV. So this is a protein that has the potential to affect delivery of the virus. On the y-axis is mutagenesis, or genetic diversification, as read out or quantified by Illumina sequencing. So you can see that with every single one of these dots, we're introducing genetic diversity into this locus. As compared to the bottom panel, which is a control guide RNA, a non-directed or non-targeted guide RNA, where there's simply background Illumina sequencing error. We then selected this virus for the ability to form more AAV. So specifically, we took the supernatant of cell culture, isolated the AAV, put it onto new cells, which enabled further genetic diversification and selection for better secretion of AAV. And after doing this for several rounds, you can see that we had significant enrichment for a particular genetic diversity, particular SNPs within the viral genome. And for example, this one was actually amplified 400-fold as a result of this selection. We then tested whether or not these resulted in improved AAV or improved delivery. And you can see here, this is the proportion, either in overall counts or in the percentage of the virus that was produced from the cell culture that was in the AAV form. Wild type, it's known, is around 0.5%. There was a known variant from literature that doubled this to around 1%. In contrast, we have a 15-fold higher amount of AAV within this particular variant that was engineered. While this was engineered in cell culture as a proof of concept, we wanted to test whether this variant had any improvement in delivery in vivo. So we ended up creating a tumor model in vivo. And the way that this was done is that now mouse colorectal cancer cells were injected into a murine model. This was followed up by administration of the virus. And you can see that with the vehicle injection or injection of wild-type vaccinia, there's not a significant difference between these in survival of the animals, whereas we see a significant increase in survival with the selected or engineered variant. And this is correlated with decreased tumor burden. We also wanted to test whether this virus was capable of traveling. In this case, we made it from a needle tip to a tumor, can it go from one tumor, a primary tumor, to a secondary tumor? So we modeled that by creating a bitumoral model and then did the injection of the virus directly into a first tumor. And so you can see here that all these versions that we tested arrived safely around the same amount inside the primary tumor. But when we then tested a later time point, whether this virus actually spread within the primary tumor and spread to the secondary tumor. And so we can see that compared to the wild type or the known variant, there's a pretty substantial increase in the spread to the other tumor. So both of these, we hypothesize, are going to result in improved efficacy as this technology advances towards preclinical development. So I'd like to summarize by saying that directed evolution is a very powerful approach to overcome problems in gene delivery. And we've used this approach to reprogram AAVs with enhanced efficiency, targeted delivery, and immune evasion. Vectors that we have engineered have currently advanced into 10 human clinical trials. The 7 that have reported data to date have all been positive, and we look forward to advancing vector products that encode genome editing machinery, specifically CRISPR, as well. And finally, directed evolution can also be harnessed as a result of CRISPR-enabled genetic diversification technologies to evolve vaccinia into an enhanced oncolytic virotherapy with better delivery to a tumor. So I'd like to acknowledge the people who actually did this work. The work on directed evolution in the mouse eye was done by Leah Byrne and Denise Delcara, each of whom is a PI in their own lab now, and this is in collaboration with John Flannery from Berkeley. The work on retrograde transport was done by Tom Guy and Bumyol Wong in collaboration with Genelia Farm. The clinical work was done by a very large team of people from 4D Molecular Therapeutics. I'll acknowledge our CEO as well as our former CSO, CMO in this slide. And finally, the work on the use of Evolver to be able to create improved vaccinia virus is the product of Huncho Lee and Adam Schaffer in close collaboration with John Duber at UC Berkeley. So thank you very much for your time and your attention.